bioconf, you have an S, that's for santé. And santé is really what we are all hoping to get our new research and inventions. So we have put together this roundtable, in fact, to discuss biotherapies between hope and clinical reality by inviting two clinicians. The Professor Philippe Menasché, who's an MD PhD, professor of thoracic and cardiovascular surgery, and he's also a team leader of regenerative medicine for cardiac and vascular diseases. He has conducted the first-in-man transplantation of skeletal myoblasts, also the first transplantation as patch of cardiac progenitors as derived from ES cells. He's also interested in paracrine effects and of the secretome of these cells and how they can affect regeneration. So he will tell us all about his work. We've also invited Professor Makoto Miyara, who's PPH, a professor in neurology at Faculté Sorbonne University and praticien hospitalier in immunology at Hospital Pitié-Salpêtrière. So he's working on the optimization of cell therapies with immunoregulation effects, in particular working in transplantation, autoimmunity, and regenerative medicine. So thank you for joining us, and perhaps we can start by discussing the hopes that you have had in your work, and perhaps Professor Menasché can start by explaining what you're doing and what you're hoping to achieve.
Well, thank you, first of all, for the invitation. What we are currently doing a clinical trial, you mentioned it briefly, using the secretome enriched in extracellular vesicles, secretome from cardiovascular progenitor cells derived from iPS. This is the continuation of the previous trial during which I transplanted surgically cardiovascular progenitors themselves from pluripotent stem cells, and as many others. We then realized that most of the effects were due to paracrine signaling, and so still use the cells, but ex vivo, just as producers of the secretome, and only deliver the secretome to the patient. So we are now doing the clinical trial. My hopes, well, you know, when you embark in this area, you're very careful. You refrain from hype because we got, as others, but we got a lot of failures and disappointments. So far the hope was that the product is well tolerated, which seems to be the case. Then the hope obviously is that it can improve function of the patients we're dealing with, that are patients with severe heart failure, but it's still a hope. It's not yet, unfortunately, a reality or a fact.
Thank you, Professor Miyara.
Okay.
What are your hopes? You're working in a particularly tricky area of immune regulation. What are you hoping for?
Yeah, that's true. So myself, so I've been working for 25 years now on those regulatory T cells. So the question was asked before about how the immune system is involved in rejection of progenitors, for example, allogenic ones. So myself, I'm highly interested in those immunoregulatory T cells that are important to prevent autoimmune response. So we can manipulate those cells to induce tolerance to organs that are transplanted, for example. And so I'm leading 3 clinical trials. That's phase 1 trial, where we isolate those regulatory T cells from patients that got kidney, liver, or lung transplantation. We expand the cells at the Miyara Center, so you got a speech from them today. And they're amazing collaborators to work with to produce those innovative therapy products. And so we have these phase 1 trials in those patients where we want to see if those cells, autologous cells, are well tolerated. So that's the first phase. And even this first phase is difficult. And it took years for us to convince people that it was first feasible, useful, and also that people should invest in those kind of treatments. So it was very tricky. And actually, we got those funding from national funding for clinical trials. And those fundings are not scaled for those kind of clinical trials you need maybe 10 times more than the highest limit of what you can get. So you need to find other ways to, to be able to run those studies. So this is the first part. And second one, now we are trying to modify genetically those cells to make them more efficient in those patients. So again, we were very fortunate to have the funding from the institutions, or public funding for that. But still, we know that it's just a first step, and it's really important to have the industry to support this kind of initiative, because basically public funding is good to start with, but when you need to get results, that's what was shown with the previous talk, you need the industry. So my hope is that the connection, that's what you do here, and also what at the APHP hospital is done, is to promote this interaction between what we do at the basic science and the clinical research, and then transform that. So that's why we need knowledge about different kind of mindset. So we are clinicians, we are scientists, but we are not very much aware of how the industry thinks. CEOs of companies, they think differently. So we need to learn from that. It took time for me to understand how they think, and to match and to convince them. So that takes a very long time. So maybe that's my hope, that we get this culture on all of sides of promoting this kind of innovative treatments.
So Professor Menasché, what are your principal obstacles that you have faced yourself in the development of your projects?
I think the major obstacles were technical. The challenge were technical. If I just take the example of the current trial with the extracellular vesicle enriched secretome, the challenges, the obstacles were what kind of filtration, which molecular cutoff for the TFF, how to qualify the potency assays, to what extent should we push exome sequencing of the donor cells and the secretome, and so on. So these are basically the technical, very basic technical questions we had to address. We tried to address them, but that was the main obstacles, and it took a little bit, say, 10 years to move on towards the clinical trial and to start in the patients. But I would say more technical than regulatory or financial.
Right. So this is basically calling for multidisciplinary teams and having a whole, different professions around you to take these complex projects all the way to,
yes,
to the patient.
And I would take this opportunity to echo what Professor Miyara said regarding Miyari, because actually both the previous trial with embryonic stem cell-derived cardiac progenitors and now the secretome, everything has been done. I mean, the production, the JMP production has been done at Miyari. And Miyari at Saint-Louis with first Jérôme Larguero, now Jean-Roch Fabreguet, this is really a superb platform and should be highlighted.
I will also go in the same direction as you say, with technical difficulties or, scientific difficulties. One of them for biotherapies, I think, is what you've evoked also, is the question of the immune response to the therapies that you're using, because most of us are still working with, where you're trying to develop, off-the-shelf therapies, allogenic therapies or things like that, or even, in the case of gene-modified cells. And we are still lacking good models to evaluate human immune responses, for instance. So that's a real big factor that slows down, I think, the development of these therapies towards patients, because we lack the ability to properly evaluate their safety before we move them on. Do you think that this is the case, or that?
Yes, and I think you mentioned something very important, that is the interdisciplinarity, which is absolutely required. That's the reason why we work together, because we need immunologists, we need cell engineers, we need chemists, and so on. So it's impossible to develop this kind of project, and it has been nicely shown on the slides of about Tree Frog, showing the diversity of the people working there. It's impossible to develop this kind of project if you are not putting together expertise in these very different areas, which at what point of time will merge together to end up in a product.
So So we have these platforms, like Miyara or like my lab or others, but another option is, of course, to look for biotechs or develop a biotech. What is the best solution, do you think?
So just to add something, thank you so much. What you said is exactly what we think in terms of development of innovative therapies, technical issues, but also limitations that need to be addressed before you start. Actually, you can have the best idea ever technologically, but that's obvious. But if it cannot be applied in a clinical grade, you just had a great idea. So yeah, that's the first thing to think about, is to connect with people that know, and that I would say that's the most important thing in a collaborative project. And of course, if you want to make a company that will produce yourself, you need to know what's possible and what's not, and also have a vision about what could be possible as well. So that's different stage and anticipation. So it's important to know knowledge and to be aware of what's going on.
So any examples of when you've been two patients and then had to go back to the drawing board to restart your research?
Constantly. Constantly. I don't think there is a barrier, a frontier between the bench and the bedside. And actually, right now, we are in the clinical trial, but we are extensively working in the lab with the extracellular vesicle and so on, because you always have something to understand. And so this is the formula, bench to bedside, but I think it's a bidirectional flux, and you should also go bedside to the bench constantly. Now, Makoto mentioned something very important, that is, it doesn't make any sense to work on this project if you're unlikely to bring it to the patient. And something I think is critically important is to navigate through the regulatory landscape by having very, very early, first day, early dialogue with the regulator, and provide them with a sort of flow chart saying, well, this is what I want to do, is it possible, what is allowed, what is forbidden, what should I do, and so on. Because otherwise, it's really loss of time, of resources, of energy, and so on. So you may have to make some trade-off with science, but when you're treating patients, which is our case, we're treating patients, not mice, the issue is not to publish in Nature. The issue is to treat the patient, even though you have to make some compromise with good high-level science. And so dialogue with the regulators from the beginning is absolutely key. And I must acknowledge, and you will comment on that our regulatory agency has improved over time. They have improved over time, and we found them more helpful than opponents.
So that's true, that this dialogue is key. My experience is that, well, I was young like you a long time ago, but I was very enthusiastic about developing those kind of innovative therapies and discussing with people from Miyara Institute. And first thing they said is, that's what you're talking about is not possible because, or this is not possible because, and so you get very negative signal at the beginning, so you get frustrated. But it's important to accept that because they are right. You cannot go further. You cannot say, okay, we'll try to do another way to overcome that, but it's not possible. So that's wisdom. They say, okay, this point will be a huge obstacle to move forward. So they know. So that's very important, and it was very helpful. So when you write your project as a clinical trial project, everything that could make your project rejected, they know. So that's very important
people with
pharmacists, very important. Yes, I agree with that.
So perhaps to finish this discussion, you're raising an important point, is that you want to treat patients, but perhaps more important, you also want to develop something that can be made available to more patients than your patients. In other words, to provide access to patients. And so that's also something that's, I think, really important in this discussion. And so I'm wondering if there are people in the audience who have specific points of view of that, how to help, making this reality, for patients to have access to medicines. I don't know if there are people who have opinions on that or want to discuss this, or perhaps in the panel. This is definitely a topic of great interest, I think, right now in the case of rare genetic diseases, where there are some therapies which are working but perhaps are not able to be developed by lack of market access. And so this is really an economic question also that's coming up around this problem, and that makes this clinical reality sometimes, fail of economic interests. So I don't know if there's any more comment on this topic. And yes, Maxime.
Yeah, so it feels to me like, and you have a very much deeper experience of that, but it feels to me like the need to have to reach affordable therapies is wide. It's not only a more optimized production scheme. It's also a more optimized QC, the regulatory understanding a little bit more what is important, what is not, the question of how is it going to be allogenic, autologous, what is even the costs around the source of the material if it's autologous. That's really a whole. It needs to be built, and it's not going to be one company, not even one country that's going to be able to build this comprehensive ecosystem to make cell therapies and not really cell therapies, but very close to cell therapies in your case, something that can fly. But when this is structured, and I just want to see what's your opinion about that, I feel like there is no scientific reason why it couldn't be cheap enough. But it's more implementation, construction of something that makes sense, in my opinion. And I would like to see, because you have much more experience, how you think about it.
I would say first stages as ours is for sure costly. And of course, what will make the price decrease is the upscaling. And upscaling means partners, industry, and investments. And of course, to be able to attract the investors, you need to have IP. So that would be one key message, that is to securing IP is very important at every stage. Otherwise, the industry is not interested. That's a very simple message I got. So maybe to decrease the price, you need to upscale, meaning increase to have partners. And I would say in France, we're lucky now. Sequence has now taken again the self-procure facility. So we now have good partners. I see them on some. So that's interesting to work here still, I would say. But yeah, IP first and the upscaling second.
Yes. I don't know whether I will precisely answer your question, but I fully agree with you. I think also it's important not to start from the product, cells or genes or whatever. What is important is to start from the disease. What kind of patients do I want to treat? If I want to treat Parkinson's patients or heart failure patients or diabetic patients, I have to consider hundreds, millions of patients. And so if I want the therapy to be accessible and not, confined to a club of happy fews, if I want it to be accessible, from the very beginning, I have to think of something simple and economically sustainable, which will attract the industry because they can expect a reasonable cost of good, a reasonable manufacturing cost, and thus propose a reasonable price and then get the reimbursement. And you have to think that all the way through. And so I'm a surgeon, so everything for me should be simple. I know that the only treatments which work, at least in surgery, are those which are simple. If If you start being too complicated, it will never work. Will work for 10 families in Europe with orphan disease. That's great. But if you want to treat Parkinson, you have to have something which can be scaled up, which can be manufactured at low cost. And this has been thought from the very first day.
And
otherwise, you will not invest, or you will not attract the investors. You know that in the industry and so on.
So you're right to point out the patient characterization, and we're hoping also that with maybe AI or more database, then you can also refine your study of patient and perhaps also really very well define your patient population. And that will help also to do the risk-benefit analysis, which is very often at the heart of where you can cut costs, because you can decide you will do this or not that. And so the other thing I want to just emphasize by finishing is you mentioned that the regulatory authorities have changed, and they have changed. And for instance, in the US, they are accepting to think in platform now. So they will regulate certain types of AAVs as platforms. You can propose a certain set of controls, and you can piggyback on that, on other products that are similar, and they accept that they are sufficiently similar so that you can say, okay, I don't need to control that much, or I know it'll be the same. So I think it's coming, and there will be a reduction, because right now we're making haute couture, one product very highly, nicely made, but we're going to make high fashion very soon. So anyway, no more question. Oh yes, Amanda.
Thank you so much for the stimulating discussion. I had a question, and maybe just I would like to ask you to tell us more about the next steps for your clinical trial on extracellular vesicles, because you succeeded to secure intellectual property, you succeeded to treat the first patients. You already did this in collaboration with an industrial partner. So if you can tell us more about what is going to be next, I would be really curious to know.
Well, the next step, to be very clear, is that this project is actually, I didn't want to talk too much about what we're doing, but the project is actually based on a collaboration between a PHP, to which I belong, and Fuji CDI in the US, which has produced the clinical grade cells, and we produced at Miyara, they produced the secretome. So the next objective is to complete the clinical trial, hopefully confirm, which is the case so far, the safety, and then, and it has started, the process has started, to partner with a big pharma or a large biotech, which will take that to the next step, phase 2, phase 3, and then market authorization, clearly. And this is driven, as you can imagine, mainly by our US partners. So this is the next objective. And Anne said something very important about AI, because actually many of the cell therapy trials have failed, not only because the product was not a good product, but because the patient population was not the right one, and because within a given patient population, the candidates were not the best ones. So to be able to prospectively identify responders versus non-responders is something which is going to be very important to increase the likelihood for a clinical trial to be positive. And this is something which the youngest generation should really rely on to avoid previous mistakes. But this is the next step. We hope we'll find one partner. If we don't find, it will be tricky, but they are looking for it.
We have hope. Well, thank you very much. I think for the sake of time, we'll close on this beautiful hope argument. Thank you very much, Philippe and Makoto, and thank you all for your attention for this roundtable.